If two brown-eyed parents can have a blue-eyed baby, what else about you is hiding in your DNA? Stay with me, because in the next minutes you'll finally see how a tiny grid, the Punnett square, quietly shapes who you are. By the end of this video, you'll know how to read any simple Punnett square, predict dominant and recessive traits, and avoid the classic mistakes students make in genetics tests and homework. Ready to decode traits step by step? Imagine this: two parents, both with brown eyes, shocked to see a blue-eyed baby. No drama, no mystery. It's just dominant and recessive alleles doing their quiet math in the background, inside every cell. Let's shrink down and see how. First, quick basics. A gene is like an instruction, and each version of that instruction is called an allele. For every gene, you carry two alleles. One from your mom, one from your dad, always traveling as a pair. Dominant alleles act like loud voices, recessive alleles like quiet whispers. If both speak, the loud one wins on the outside, but the quiet one can still be carried, hidden, waiting to reappear in the next generation. Geneticists use uppercase letters for dominant alleles and lowercase for recessive ones. So B might mean brown eyes, while B means blue eyes. Your combo, like BB or BB, is your genotype, the recipe behind your traits. Here comes our secret weapon, the Punnett square. It's just a simple box that helps us track all the allele combinations possible from two parents. No magic, just organized crossing of options in a neat visual way. Let's say brown eyes are dominant, blue eyes are recessive. A brown-eyed person might be BB or BBA. A blue-eyed person must be BB. To predict children, we put the alleles from each parent along the sides of the Punnett square. Example 1. Both parents are BB, brown-eyed but carrying blue. Put B and B from one parent on the top, B and B from the other on the side. Now fill the boxes by dropping alleles down and pushing them across. When you fill it, you get one BB box, two BB boxes, and one BB box. That means about 75% of kids will show brown eyes and about 25% will show blue eyes. But all four got their alleles by pure chance. Important detail: Punnett squares show probabilities, not guarantees. 4 children don't have to fit the 3-1 pattern exactly. It's like rolling a 4-sided die over and over. The odds guide us, but randomness rules each roll. Now switch traits. Think of freckles. F might be dominant freckles, F recessive no freckles. A freckled parent could be FF or FF. If they have kids with an FF parent, the square instantly shows who can have freckles. Let's do that cross. One parent FF, the other FF. Put F and F on the top, F and F on the side, then combine. You'll see half the boxes FF and half FF. So around 50% freckled, 50% no freckles in their children. Notice the pattern? Dominant traits only need one copy to show. Recessive traits need both alleles to match. That's why recessive traits like blue eyes or no freckles often skip generations and pop up unexpectedly. Now, many students confuse genotype, the allele letters, with phenotype, the visible trait. BB and BB are different genotypes, but they share the same phenotype: brown eyes. Only BB looks different on the outside. Another trap. People think Punnett squares predict a single child. No, they model the chance for each child independently. So every new baby is like another spin of that genetic wheel, using the same probabilities. Punnett squares also help explain carriers. A B person looks brown-eyed but carries the recessive blue allele. 2 carriers can have blue-eyed children even if neither parent shows that trait at all. It's hidden in the letters. Let's shift to plants where these ideas began. Picture pea plants. Tall is dominant, short is recessive. A tea plant is tall but carries short. Crossing 2 plants gives that classic 3 tall to 1 short ratio. Fill that TTTT square, you get TT and TT. 3 tall phenotypes for each short one. This pattern was exactly what early scientists like Mendel saw when they counted huge numbers of pea plant offspring. Dominant does not mean better or more common in the whole population. It only means that one copy is enough to show the trait. Some recessive traits can be rare, but others can be surprisingly common in certain groups. So how do we build your own Punnett square? Step 1. Decide which trait you're tracking and choose a clear letter for its allele. Capital for dominant, lowercase for recessive, making sure they look distinct from each other. Step 2. Write each parent's genotype as 2 letters, then place one allele on each side of the square, one parent along the top, the other parent down the left side. Now your grid is ready to be filled. Step 3. Fill in each box by combining the top allele with the side allele, like multiplying entries in a times table. When you're done, count the different genotypes, then translate them into visible phenotypes. To keep your brain engaged, try predicting the outcome before you draw the grid. Ask yourself, how many dominant alleles could each child receive? How many recessive? Then use the Punnett square to check your guess. Punnett squares can grow beyond 2x2 for traits with more alleles, but the core idea stays. List possible gametes from each parent, then combine them. For now, mastering the simple 2x2 will already boost your genetic skills. Think about your own traits: dimples, attached earlobes, tongue rolling. Many follow simple dominant recessive patterns, at least roughly. With a paper and pencil, you can sketch Punnett squares to explore family possibilities. One last reminder: Punnett squares do not decide your fate. They're just a map of probabilities. Real genetics is influenced by many genes, environment, and chance, but this tiny grid is a powerful first step into that world. If you learned something new about dominant and recessive traits, imagine how much more there is waiting in genetics. Hit subscribe, share this with a friend, and tell me, which trait would you love to decode with a Punnett square next?